Hi everybody. So this week, this class and the next class are going to circle around the idea of protein synthesis. And that's going to be our entry 13. So kind of looking at what we learned before, last class we focused on, okay, what does DNA look like? And what are the parts of DNA? Because it's our blueprint, right? All of our genetic material that code and make us us, it's all there. So then how do we go from having, that DNA code to actually making all the proteins that do the heavy lifting in the work? And so that's where protein synthesis comes in. Synthesis as a word means to make. And so today we're going to learn about that process and how we go from the DNA code to the proteins that do their job. And so today's goal is to just kind of finish up our notes so that when we return to class next time, we can really focus in on an activity related to protein synthesis. So I'm just going to kind of click into this presentation. And I'm going to open it. If you're not able to be there with class, then you can kind of go through this on your own. But the activity I'm hoping to do next class is an escape room activity. And this is kind of an introductory version that does not require you to know the whole process of protein synthesis in order to do. It has you remember the idea of decoding from last class. So what you're going to do is I have a special code chart that you are going to follow. And I have a couple of rules that I'm going to introduce to the class. Every 3 letters of code that I present you is going to correspond to a particular letter. Similar to Morse code that we talked about last time, that was a series of, dits and das or dots and dashes, however you want to remember it. In certain combination and order, they produce different letters. Well, here, every 3 letters is going to represent of the code will correspond to an English language letter. And we're going to use that chart on the right to help us. So to figure it out, you first pick the first letter on the left-hand side. So if the first letter is a U, you're going to come up here in this row. If the second letter is a C, you're going to be in the second row, A and G. Then you kind of come up to the second letter and the third letter. And that's just to help guide you. You can clearly see in the chart, like you don't necessarily need to follow the 1, 2, 3 letter pattern. It just helps you find them faster. But once you've successfully decoded the message, you are going to check your answer on the Google form. And I will have a Google form linked here. It's not for whatever reason. I think it's actually on the next slide to see if you can move on. It'll say try again if you get the wrong answer, or it'll ask for the next thing if you get it correct. After the third message is decoded, you're going to get kind of a victory phrase or a code that your job is to give to the teacher. Okay, so the next series of slides just has the different message. So you're going to use this code, and I tried to help you a little bit to get you started. I've broken it into the 3-letter chunks. Then when you think you have it, you're going to go to the Google form, which I can just click open right now, and you're just going to type that message in. If I just type any old random thing and try to go on, it's going to tell me that I'm not correct. You're going to have to actually get it correct in order to move on. When you do, you'll get a second message, you'll check it, and then you'll get a third message, which you actually have to separate and decode. And if you do all of those things correctly, you are going to get a victory phrase that you will bring back to your teacher. So that's just a fun little intro decoding activity that I plan to do with the class in breakout rooms, in small groups, just to kind of get you warmed up to the idea of decoding. And then we're going to launch into entry 13. So our essential question, make sure you pause, open up to a next fresh page in your journal, and try to write down how are proteins made from the genetic code in our DNA. So again, pause, make sure you get that down. And then we'll launch into how to set up our A page. Okay, so we have a lot to cover today, and I just want to help kind of guide how your notes are going to go today, just to kind of make sure that you have enough space in what you're going to do. So you might not know any of these terms on the table yet, but you will by the end of today. So we're going to kind of create a table that has one longer rectangle up top, and we're going to put in there something called the central dogma of biology. And then we're going to talk about the two pieces or two processes of protein synthesis. One of them is called transcription and one of them is called translation. So we're going to kind of have two boxes for one and two boxes for the other. So please pause the video and take a second to kind of set up your page, or at least remember how I broke it up, so that way as you're copying down your notes, you kind of have the gist of what should go where. Okay, so kind of also thinking back to what we've learned last time, and you don't have to write this part down, I just want to give you a frame of reference, right? DNA is inside the nucleus. And that is where all of our genetic material, all of our blueprints live to make everything we need for life. Think of the nucleus like a bank vault, right? It is keeping DNA safe and secure inside. But the trick is, the DNA can't leave. It's trapped in there. So how do we get proteins that go and do their thing all over the cell and even leave the cell and go do things in other places if the DNA itself is trapped inside the nucleus? Enter that central dogma of biology. So at the very top of that note sheet underneath your essential question, I do want you to pause for a moment, and I want you to draw out this picture in that area. If you'd like, you can write the text also, but the picture is kind of the most important, and we'll circle back to it at the end of the presentation as well. But the key hallmark piece is that the processes of transcription and translation are the two processes by which DNA that's trapped inside the nucleus can actually communicate all of the instructions to make proteins, which, as we know, can give us the traits that we have. So we're going to be introduced to something today called mRNA, which we've never talked about before, but it's kind of that intermediate, and the M stands for messenger, as we'll find out. So it's the messenger that kind of carries that DNA message to then make proteins. And that's just kind of the central dogma of biology. DNA will go to mRNA, which goes to proteins in a step-by-step process. All right, so first up, we're going to talk about transcription. And this is where I would like you to write the definition and/or any word notes kind of in that middle left box on our A page. Transcription, if you've ever transcribed anything, it means to write. So transcription is writing down a copy of the DNA code. Because DNA is trapped inside the nucleus, this takes place in the nucleus, and we are creating or copying a gene, which is a chunk of DNA that codes for a protein. So we're not copying the entirety of all the DNA. We are kind of zooming in on one small chunk called a gene, and that little segment will eventually code for one protein. So we kind of unwind and unzip that double helix of DNA, which we'll talk about in a second, and make a copy. We write down one copy of that gene in the form of messenger RNA. So pause, take a second, and kind of write that text piece down in that box so you have it on your notes. And you don't necessarily have to write this piece down. We're going to end up drawing a picture of it. So just kind of follow along with the notes, and I'll tell you when really to write something down. But if we remember, DNA is a twisted helix that's glued together by hydrogen bonds, right? The A's and T's and C's and G's are stuck together by hydrogen bonds. So in order to actually like get to the DNA to make a copy, we have to unzip it. So there's an enzyme that comes along and it basically snips all those hydrogen bonds and peels open the DNA. And now there's access to it. And then from there, we can actually make the mRNA copy or messenger RNA copy of that gene. And to do this, we follow the same basic pairing rules that you learned with DNA. C's are always going to go with G's. And normally, right, we learned A's always go with T's. However, there's a trick. In RNA, they have something different. RNA does not have T's. They have U's, which stands for uracil. So it's still a 4-letter code. However, everywhere you would have maybe wanted to put a T if it had been DNA, instead you just swap it out for a U in RNA. So if the DNA is a T, it'll still pair up with A. If the DNA is an A, instead of pairing it with a T, we pair it with a U. And C's and G's will still go together. So you can kind of see how the RNA in red here is being created. A, U, G, A, U, C, U, and so on, all the way down kind of to the end of the gene. And it's important to realize that unlike DNA, messenger RNA acts as a messenger, so it can leave the nucleus. So that's really why it's a messenger. It takes the message that it just created or copied from the DNA and exits the nucleus. And it has all that gene making information with it. So I do have a little video recap here. If you click on it, it'll bring you to a nice little refresher video on transcription and kind of is only a few minutes long and does a nice job of highlighting all of the steps. So please take a moment and maybe click on that and watch it so you have a good idea before moving on. All right. So what I want to do is try to draw this on your A page. And in a perfect world, I would have, given you a printed sheet of paper and you just color and that's it. But here we are. So in that bottom left space, we're going to draw a picture. So I kind of put a photo of my hand-drawn one here, but I'll give everybody like, 10 minutes in class to draw this. But some of the homework things that I did, I have the nucleus, which I kind of drew in this bluish color here, and it does have pores. And then I drew my DNA. You can see it's that twisted ladder, and then it gets opened up where the message is going to be copied. I tried to color code throughout, so I have my gene, my DNA gene here in green. I also copied the text on the left. So that message that you see here on the left in green is the same exact thing if you read from top to bottom, the green message on my picture. And then you can see the mRNA code is made from that DNA message. And you can see it follows the base pairing rules. And then everywhere you normally would have a T, instead you have a U. And that's one easily distinguishable feature between DNA and RNA. You will never find a U in DNA, and you will never find a T in RNA. All right, and then as you can see here, I show that mRNA is now leaving the nucleus. There's a little arrow that kind of got cut out of the photo here that's going to lead into the next part of the picture a little later. So that's the key piece of transcription, is writing down that message of the DNA gene into mRNA. So now we have to ask ourselves, that message now left the nucleus. Where is it going? Where in the cell are proteins made? We learned this in our cell unit. They go to the ribosome. The ribosome is the site of protein synthesis. So here in our text part of translation, I want you to pause and write down this text. So translation is the making of a protein from an mRNA template. So we have the mRNA message. Now we're going to actually take that info and translate it into a protein. And really it's like Google Translate, right? You pop in one language, you get out another. In this case, we are officially translating the language of nucleic acids, or DNA, into a language of proteins. And again, this happens in the ribosome. Okay, so another thing that kind of points to what we did at the very beginning of class today is that mRNA is a code, right? It is read in 3-letter words, so to speak, called codons. So similar to what we opened class with when I had 3-letter chunks that had A's, U's, C's, and G's, you didn't know that it was mRNA at the time, but it was. Those are called codons. So you can see I've kind of broken them in 3-letter chunks here. To kind of give you an idea, there are 4 codons in this tiny little message. And each one of those codons, or 3-letter codes, codes for one particular amino acid. We got to think way back, but amino acids are the building blocks of proteins. We string them all together and we get a protein. Oops, sorry. There are 20 different amino acids to choose from. And so that combo is kind of another code. There are 20 amino acids, and when we put them together in certain ways, we can get more and more different types of proteins. And so, like I said, just kind of hanging out in the cytoplasm or the open space of the cell, there are all of these amino acids. And there's something called tRNA. Sorry to burst your bubble, there is more than one kind of RNA that exists. So we've already talked about mRNA, which is the message RNA that carried the message from DNA outside the nucleus. Well, we also have something called tRNA, and the little t stands for transfer, and it does exactly that. It transfers the correct amino acid to the corresponding code or codon on the mRNA. It kind of just matches them up. Oh, I guess I should kind of see. So basically what it does is it acts as a little taxi. So think of tRNA as transfer RNA or taxi RNA. There are specific taxis for specific amino acids, and they will only recognize specific codons. So these little transfer RNAs are floating around with their amino acid, and when they recognize their 3-letter code, they will zoom in and drop off their amino acid and then go back and find another. And they recycle themselves. And then it will go grab another, bring it in, grab another, bring it in. But it will only come in and drop it off to its corresponding sequence. It's not just going to drop amino acids in any specific order. It's reading that code in 3-letter words. Okay. And here's kind of another representation of the chart that we opened class with. I had given you the 3-letter code table at the very beginning of class, and if I scroll up, I should have left it open, but I didn't. I had given you this special decoder chart, and you can see they are in 3-letter codons, and they correspond in this case to an English letter. But in reality, they code for amino acids. So let me go back to our slide here. And this is kind of our codon chart to help us read and determine the amino acids. There are 20 amino acids, and you can see that they're written around the outside. So alanine, valine, arginine, serine, lysine, all of those are different amino acids. Methionine is known as the start amino acid, just because all proteins start with that amino acid. It's kind of a hallmark, and scientists know that, oh, there's a new protein or a gene code if it starts with methionine. There are also specific ones that say stop. Because if you're wondering, like, how do you know when you've hit the end of a protein? There's a special little stop sign signal. And so when a protein is being made, it kind of just keeps chugging along until it says, whoa, nope, you've reached the end. We're done now. And that tells the ribosome to say, hey, protein is done being made. And so there's special codes for that too. But to read this thing, we start in the center and work our way out. So for example, methionine is AUG. So you start in the middle and work your way out for that 3-letter code. And you will notice that there are some that have more than one option. So GUU is valine, GUC is valine, GUA is valine, GUG is valine. So there is some overlap where multiple 3-letter codes could correspond to the same amino acid. But then there's ones like methionine where it's very specific. So for example, let's take our 4-codon message. And I want you to use the chart to see if you can determine what those 4 amino acids would be. So pause for a minute, see if you can decode those 4 codons, and I will tell you the answer in a moment. All right, so what you should have gotten is the first one is AUG, which we talked about, methionine. And you might not know the 3-letter abbreviation, totally fine. Next up, we have AUC, which is isoleucine. The blue one is UCG, which is serine. And then we have UAA, which is a stop. Please know that amino acids in real life are much, proteins sequences are much longer than this in real life. But this is just kind of an example. So that's how we decode the message. And there would be a transfer RNA that has a methionine that would kind of recognize AUG, come in, drop it off. There would be a different transfer RNA that's carrying isoleucine that recognizes specifically AUC that would come in and drop it off. Same thing with serine and same thing with a stop. It doesn't have an amino acid, but it does kind of have its own special signal. So this is kind of a picture here you can kind of see on the right of the ribosome. It's got 2 parts. The mRNA message is kind of sandwiched in between those 2 big parts. And again, it's got the message in 3-letter codon words. And then tRNA floating around will read that message and add on their amino acid if it matches. And you start to build these little circles, which are each one is an amino acid, and then you start to link them all together, and you get that crazy 3D structure, and voila, you have a protein. And we have learned about proteins before, right? We talked about structure determines function. We talked about how they act as enzymes. They pretty much help us with all of our different reactions in our body. And they give us the traits that we have as well. Similar to transcription, there's a little video recap. If you click on it, it'll take you to a couple-minute video that kind of puts it all together for you. So please take a moment and watch that as well. Okay. So in your notes, here's kind of what my photo or drawing ended up looking like, and you're going to copy that onto the bottom right of your notes page. You can see where I had drawn transcription, where I had my mRNA leaving the nucleus. Well, then it goes to the ribosome for translation. So just kind of articulating what you see here. My ribosome is kind of in dark blue. You can see that my mRNA message is in there, red in the 3-letter codon chunks, and each codon codes for an amino acid. You can see how there's tRNA out there in the cytoplasm along with amino acids. And they also have what's called that anticodon. That's what registers the message. That's how it's super specific. So if there's a tRNA that specifically recognizes AUG, it will come in and drop off the amino acid. And so you could see we had methionine, isoleucine, serine. So you kind of get an idea. And I want you to pause and see if you can kind of recreate the process of translation onto your notes sheet. Okay, so putting it all together, how do we go from our DNA code to a protein? We start with DNA, and then we copy or transcribe an mRNA message, and then we translate it into the language of proteins by the process of translation. And the messenger RNA is the thing that actually escapes the nucleus and goes to the ribosome to make the proteins. I have a nice little kind of GIF here that kind of shows you the different steps. You don't have to know the specific names of any of the enzymes or anything, but you can kind of see what's happening in transcription and translation. Each one of those green guys is a tRNA dropping off its amino acid. Okay. And that brings me back to the beginning of this presentation at the top of your notes page where you have the central dogma of biology. So we had learned about DNA in a gene, and then it gets turned into RNA by the process of transcription, gets turned into a protein by the process of translation, and then those proteins give us our traits. So we kind of wrote this down without totally knowing what was going on, but now we have kind of an understanding of how we get to each one of those steps. Here's a picture of my notes page. If it's helpful, I apologize, it's kind of dark, but you can kind of see the two pictures that I drew. You can see the central dogma up top and my word versions of those two processes there as well. Okay. There is a really good interactive where you actually have to go through and click on things to practice. So I'm going to encourage the class to make sure that they try this. If you click on it, it'll take you there. You just kind of click on it to start, and it'll walk you through the process. You might have to watch a
Transcription and translation are the processes that all living things use.
A short video, and once you walk through the short video, it'll prompt you to select a couple of things and choose and do a couple different things. So it's kind of fun. It's a really nice recap that brings everything kind of back together on what we have learned today, because it is a lot of information. And this is where class is going to stop, and we'll pick up again next time. So thanks for listening.